Hi, my name is Ariel Madrigal, and today I'm really happy to talk to you about our project, which is called a unified model for Bayesian integration and interpretation of single-cell RNA-seq data. One of the problems that exists in the analysis of single-cell data is a problem known as integration. What we are facing here is that we have unwanted sources of technical variation present within our dataset. In this case, represented by the presence of multiple batches. When we generate, for example, a dimensional reduction embedding of our data, what we might observe is that instead of having clusters that represent the cell type of origin, we have clusters that represent the batch. In order to solve this problem, a lot of different computational methods have been developed over the recent years, with the idea of removing these unwanted sources of technical variation and preserving the original cell type identities. However, more complex scenarios might arise. For example, we might observe a situation that looks something like this. The idea is that we have the same scenario as before, but now with the presence of two biological conditions of interest. For example, condition 1 might represent cells profile from a healthy donor, while condition 2 might represent cells profile from a donor with a disease. What we want to do is that we want to learn the differences between the two biological conditions of interest, while also removing unwanted sources of technical variation. Therefore, the problem is that we want to decompose the different effects of sample-to-sample variability that exists within our dataset. In order to tackle this problem, we have developed GEDI, which stands for Gene Expression Decomposition and Integration. And before I go into the details of GEDI, I just want to mention that a lot of the methods that were developed for integration have this underlying assumption that they want to align the manifold of multiple samples. So what GEDI is doing is that it creates a reference frame defined by these set vectors. This reference frame defines a manifold, and here exemplified by this ellipse. In this manifold, different cell types of population, such as the red and the blue areas, may lie in. We can project the transcriptome of a given cell into this manifold. Then, when we analyze a given sample, we can quantify the changes that this specific sample would have to the reference frame. We might realize that different samples might have different distortions to the reference frame. Finally, we can connect the changes of the reference frame to sample level variables. GEDI can also be used to model the manifold as a function of gene level variables. And what this means is that we can use prior information about pathway membership, gene regulatory networks, or cell type signatures in order to guide the creation of our reference frame. GEDI can work with different types of data distributions. So it can work with either with normalized data, with raw count data, or it also has the ability of modeling the ratio between two entities. This specific capability is really useful for the analysis of the splicing and or stability. What we wanted to do first was to measure the ability of GEDI to capture sample-to-sample variability. To do this, we looked at this data set that profiled different immune cell types across multiple single-cell RNA-seq technologies. What we first did was to look at the changes that each individual sample would have to the reference frame, and we visualized these changes with UMAP. So what we have in this plot is that each dot represents one sample. In this sample level analysis, what we are able to see is that we can recover variability associated with each donor while removing variability associated with the different technologies. We can kind of repeat the same analysis, but now looking at the single cell level. This plot represents the integrated space after running GEDI. And what we are able to see is that we are able to recover the different cell types that exist while removing variability associated with the different technologies. we can not only visualize this, but actually quantify this change, this performance with this overall score that again is a measure of how well we are able to remove the technical variability while preserving biological variability associated with the cell types. In our benchmark, GEDI stands out as a top performer against other integration methods that have been developed previously. Having established that we can capture sample-to-sample variability with GEDI, now what we wanted to do was to actually model sample-level variables using GEDI. One way in which we can quantify the changes that would occur to changes in sample-level variables is through these transcriptomic vector fields. So the concept here is that, again, we have our reference frame defined by their set vectors, and what we are measuring are the changes that would occur to the reference frame given a change in a sample-level variable. So these changes represent the vector field, and although it's multidimensional, we can visualize it as arrows in a two-dimensional embedding. One way in which we could, we wanted to now look at a data set that had different conditions of interest. So we looked at this data set, which is a collection, again, of different immune cell types profiled from 40 donors, in which we have 3 biological conditions of interest. We have control, mild, and severe COVID-19 cases. So the first thing that we do is that we use GEDI, and we are able to, again, generate an embedding that recovers the cell type structure of the data. But now we want to learn something about the differences between our disease groups. We want to learn what would be the changes that would occur between the severe and the control conditions. We can visualize these changes with our vector fields, which are a cluster-free way of visualizing the gene expression changes that occur. So these vector fields are telling us what will be the changes that would occur for a given cell state going from the control condition to the severe COVID-19 condition. We can see that certain cell types have cell states really enriched, for example, really enriched for gene expression changes. For example, the monocytes, neutrophils, and also some populations of T cells. We also wanted to see how well this approach compares to previous approaches. So what we did is that we performed a pseudobulk analysis, and then we took the mean vector field per cell type, and we compared it to the pseudobulk approach. What we observed is that our gene expression changes were really concordant with a pseudobulk approach. We can also use GEDI to model the manifold as a function of gene-level variables. And what we're doing here is that we are using prior information about regulatory networks. The concept here is that in an embedding, we can project the activity of a given transcription factor, and we can calculate the gradient, which is exemplified by this arrow that goes across the diagonal. Then we can compare the gradient vector field versus other transcriptomic vector fields to see if they are pointing in the same direction, and they both have high magnitudes. Again, we look at our COVID-19 data set. So we have an embedding showing the different cell types. But now what we wanted to do was to look for transcription factors that were, again, pointing in the same direction as the vector field of severe COVID-19. So we identified this transcription factor called SPI1. So we first project the activity of this transcription factor. And from here, we can calculate the gradient vector field of this transcription factor activity. And then what we can do is also compare it to the vector field of the severe COVID-19. What these plots are telling us are that in the monocyte population, there seem to be high gene expression changes that occur for this cell state in the severe COVID-19 cases, and that the SPI1 activity is also correlating and pointing in the same direction as the vector field of severe COVID-19. GEDI can also be used for the analysis of ratio data. What GEDI does in this case is that it models the log-odd ratio of the reads that would support an exon inclusion event versus the reads that would support an exon exclusion event. We use this data set of mouse neurogenesis from 2 different studies to quantify the ability of GEDI to perform integration of the exon inclusion and exon exclusion counts. What we observe is that GEDI is able to remove variability associated with the different studies and is able to recover the different cell types. When we quantify the ability to remove technical variation, we observe that GEDI stands out as the top performer. And again, this emphasizes that GEDI is the only method available to model the ratio of 2 entities. And finally, we are able to identify novel exon inclusion events that are cell type specific. So in summary, GEDI decomposes the effect of different sources of sample-to-sample variability. GEDI learns cluster-free gene expression changes along a continuum of cell states and reconstructs COVID-19 associated vector fields. GEDI identifies transcription factor whose gradient correlate with COVID-19 associated vector fields. And GEDI allows the study of alternative splicing landscapes, revealing cell type specific cassette exon inclusion events in mouse neurogenesis. With this, I just want to conclude and thank you so much for your attention.